Welcome to this explainer. Today, we're breaking down a course you might be gearing up for, Fundamentals of Biosciences, officially known as UCHP 112. Now, if you're stepping into this class, you're probably looking for a roadmap to navigate what feels like totally new terrain, right? Well, that's exactly what we're going to do. We'll map out exactly what you'll learn, why you're learning it, and how you're going to be evaluated by the end of the term. Think of this not just as a quick syllabus review, but really as the starting point of an intellectual journey into some of the most complex chemical and physical systems we know of.
So right off the bat, we've got to address the elephant in the room for first-year hard science students. Why exactly do chemistry and physics majors need a biosciences class? I mean, it's a completely fair question. You signed up to study quantum mechanics and thermodynamics or, organic synthesis. Yet, here you are staring down a biology curriculum. It might feel like a detour, but there is a fascinating, highly practical reason this is a core requirement for your degree. The boundaries between these scientific fields are dissolving faster than ever before.
The official course description actually gives us a massive clue here. It explicitly says the course emphasizes interdisciplinary applications of biosciences relevant to chemistry and physics, such as diffusion, osmosis, enzyme activity, microscopy, and analytical techniques. This curriculum brilliantly bridges the gap. It focuses heavily on the chemical and physical mechanics that literally drive living organisms. Modern biology, honestly, isn't just about memorizing taxonomy anymore. It is applied physics and applied chemistry playing out in the most dynamic systems on Earth. Concepts like diffusion and enzyme activity. Those are the ultimate crossover events where your core major directly dictates how life survives.
To help you master this material, here's our game plan. We've got 5 key stops: biology for hard sciences, mapping the plant kingdom, animal diversity and physiology, the experimental bioscience lab, and finally, course mechanics and evaluation. Let's dive in.
Okay, Part 1, Biology for Hard Sciences. Let's look at the actual course objectives.
The syllabus outlines 5 specific course outcomes, or COs. Consider these your ultimate checklist for success. By the end of the semester, you need to be able to classify major plant and animal groups, illustrate their structural organization, and compare basic physiological processes. But pay special attention to outcomes 4 and 5. This is where your specific background really shines. You're going to apply concepts like diffusion and osmosis to biological experiments and identify applications of microscopy and bioinformatics. Imagine a physicist using advanced microscopy to track cellular movement, or a chemist analyzing the exact catalytic rate of an enzyme inside a living cell. You aren't just observing biology, you're decoding it using the rigorous tools of chemistry and physics.
Moving right along to Part 2, Mapping the Plant Kingdom. Let's talk plant diversity.
For the first 12 hours of your lectures, you'll be going on a massive evolutionary journey through modules 1 and 2. You'll start with simpler organisms like algae and mosses and track their development all the way up to complex seed-bearing gymnosperms and flowering angiosperms. But you won't just look at the plant as a whole. You'll study functional biology. This means really digging into the mechanical functions of roots, stems, leaves, flowers, and seeds. You're essentially studying the biological engineering that allows a completely static organism to pull nutrients from the earth and energy from the sun. Pretty wild when you think about it.
Now, you might be wondering, how do we actually know how all these diverse plants are related to one another? Well, that brings us to phylogenetic analysis. You'll dive deep into this concept, discovering how molecular chemistry is used today to map out the evolutionary history of plants. We have moved way beyond just looking at the shape of a leaf to classify a species. Modern taxonomy is incredibly sophisticated and heavily relies on DNA-based molecular approaches. For the chemistry students out there, this is right up your alley. You're looking at the foundational macromolecules of life, the sequence of base pairs to trace millions of years of evolution. Taxonomy today is basically applied molecular chemistry.
All right, Part 3, Animal Diversity and Physiology. Let's shift our focus to comparative biology.
Here is where we build a powerful comparative perspective between flora and fauna. You'll contrast the structural tissues of plants with the highly dynamic systems of animals. Think of it like this. Plants utilize meristematic tissues, which are basically plant stem cells that allow for continuous growth and amazing feats like vegetative propagation, where you can grow a whole new plant from a simple cutting. Animals, on the other hand, are highly active. They don't just stay in one place. They move around, they hunt, they react. And that requires robust, interconnected physiological processes to survive. They rely on complex internal systems that operate like highly refined biological engines. Module 4 expects you to build a foundational understanding of these specific physiological processes. We're talking about the nuts and bolts of digestion, respiration, circulation, excretion, and reproduction. You'll study specific animal tissues and their functional significance, but I want you to think about this from a hard science perspective. Digestion is basically a series of complex chemical breakdowns and enzymatic reactions. Circulation, it relies entirely on fluid dynamics and pressure gradients. How do the tissues in a respiratory system physically and chemically facilitate the exchange of gases across a membrane? That intricate intersection of physics and chemistry, that's the exact level of understanding you need to reach.
To really wrap your head around these vast systems, biology often scales from the macro down to highly specific examples. So you'll be introduced to representative animals, the earthworm, the cockroach, the frog, and the fish. And look, these aren't just random creatures you'd find outside. They are the foundational blueprints used by biomedical researchers all over the world. By understanding the organization, internal anatomy, and evolutionary trends in these specific model organisms, you gain the context that scientists use every single day to understand complex human physiology, test new life-saving drugs, and unlock deep genetic secrets.
Section 4. The Experimental Bioscience Lab. Time for some experimental biology.
If you're someone who loves being at the bench, Module 5 is absolutely for you. This is where theory finally meets practice. You're going to learn the physical principles of microscopy, which is pure optics and physics, and the chemical principles of staining cells so they can actually be seen. You'll apply concepts like enzyme activity, photosynthesis, and osmosis in real hands-on experimental designs. And most importantly, this module reinforces the absolute bedrock of the scientific method, mastering controls and variables in your experimental planning. This is the universal language of all hard sciences, whether you're splitting an atom, synthesizing a compound, or tracking Mendelian inheritance in a petri dish.
A massive highlight of this module, and honestly, a real game changer for any modern scientist, is bioinformatics. This introduces you to the massive biological databases that are completely revolutionizing how we analyze living systems. We are talking about the intersection of big data, computational algorithms, and biology. The sheer scale of data generated by modern genetic sequencing is staggering. Bioinformatics shows you how these massive amounts of molecular data are stored, retrieved, and modeled by computers to find patterns that the human eye could literally never catch on its own. It's computational physics and data science mapped directly onto life itself.
Finally, Part 5, Course Mechanics and Evaluation. Let's talk grading and logistics.
Okay, let's talk about the actual logistics of the course. This is a 2-credit theory course, demanding 2 hours of your time per week. The 50 total marks for the course are divided into 2 distinct categories, which is great because it ensures you're continually keeping up with the material rather than just panic cramming at the very end of the term.
So, your final grade is a combo of your in-semester continuous assessments, which total 15 marks, broken down into a 10-mark and a 5-mark component, and your end-semester exam, which carries the real weight at 35 marks. It's a very straightforward, balanced approach to tracking your progress.
But here is the absolute most important number to remember today, 40%. That is the minimum passing threshold you must achieve. But, and this is a critical caveat, you must achieve that 40% in both the continuous assessment portion and the final end semester exam. You can't just ace the continuous assessment and bomb the final, or vice versa. Consistent effort across both evaluation phases is required to secure your 2 credits and pass the course.
The syllabus provides a fantastic set of references to guide you along the way, featuring authors like Gupta, Singh, Purohit, and Verma, covering everything from basic cell biology to the expansive horizons of biotechnology. You have the structural blueprint, and you know exactly how your hard science background perfectly equips you for this material. So I'll leave you with this final thought. If biology is ultimately just chemistry and physics scaled up into incredibly complex, living, breathing systems, what previously unsolved physical or chemical mysteries might you finally unlock by studying life itself?